Λοιπόν, μαζί μας, όπως σας είχαμε μιλήσει, έχουμε κάποιους εκλεκτούς ομιλητές, τους οποίους θέλουμε να αφιερώσουμε περισσότερο χρόνο για να μας μεταφέρουν την τεχνογνωσία τους και θα ξεκινήσουμε από τον Αλμπέρτο, να βρω τον Αλμπέρτο, τον Alberto Urbaneja, που είναι ερευνητής καθηγητής εντομολογίας στο Ινστιτούτο Γεωργικών Ερευνών της Βαλένθια στην Ισπανία, αλλιώς Ήβια γνωστό. Καθ' όλη τη διάρκεια της καριέρας του έχει ασχοληθεί με πολυάριθμα σημαντικά εντομολογικά προβλήματα στις καλλιέργειες, εστιάζοντας συστηματικά στην ανάπτυξη προγραμμάτων διαχείρισης επιβλαβών οργανισμών με προτεραιότητα και πάλι το βιολογικό έλεγχο. Η έρευνά του αφορά καινοτόμες στρατηγικές για την ενίσχυση της ανθεκτικότητας των φυτών, αξιοποιώντας φυσικές οικολογικές αλληλοεπιδράσεις. Από τα πολλά έντομα εχθρούς που έχει μελετήσει, τα επιβλαβή έντομα των εσπεριδοειδών αποτελούν έναν από τους βασικούς ερευνητικούς του άξονες. Και αυτός ο ρόλος του φέρνει τον Αλμπέρτο στην Κύπρο. Έχει εργαστεί εκτενώς πάνω στη διαχείριση τόσο της αττικής ψήλας των εσπεριδοειδών, όσο και σε άλλα επιβλαβείς παρόμοια έντομα, που είναι φορείς της ασθένεια του βακτηρίου που προκαλεί την ασθένεια του κίτρινου δράκου. Πιο πρόσφατα έχει αναπτύξει βιοτεχνολογικές στρατηγικές για την ενίσχυση της αντοχής των εσπεριδοειδών στο στρες μέσω της ενεργοποίησης αμυντικών μηχανισμών των φυτών και έχει συντονίσει και συγγράψει πάνω από 400 δημοσιεύσεις και ένα τεράστιο αριθμό ερευνητικών έργων. Θα μας παρουσιάσει τώρα το έργο του. Αλμπέρτο, αν μπορείς να έρθεις στο βήμα για να ακούσουμε από σένα τι έχετε κάνει το έργο σας όσον αφορά την ασιατική ψήλα. Ευχαριστώ, Αλμπέρτο.
Thank you, Antemis, for the introduction. And before to start, obviously, I have to thank the Department of Agriculture of Cyprus for all the collaboration. And as you will see in the presentation, for us it's very important to be here and to assist in all what we can assist. Okay.
Translation because
there is
feedback.
Okay. Here you go. And back and okay. Okay. So I'm coming from Spain. In Spain, as you can see in the picture, we have all the Mediterranean and part of the Atlantic coast, plenty of citrus orchards. We have around 300,000 hectares of citrus, and we are the main producers of citrus in Spain. We are the first exporters of citrus for fresh consumptions worldwide, and with these numbers you can understand why it's important for us to deal with HLD and with Diaphorina citrus. We have, I would say, a good biological control program already established in our orchards, which is based mainly on conservation biological control. And the most important thing for us at the moment is that we are free of Diaphorina citrus, we are free of Triotha erythrae, I will explain you why, and obviously of the HLB and citrus greening. Well, I'm coming from Valencian Institute of Agricultural Research. My research institute is located in the center of the citrus production in Valencia region. Valencia region is the main producer of citrus in Spain, a little bit less than 200,000 hectares of citrus, all of them for fresh consumption. I would say 30% of clementine production, 60% of oranges, and 10% of lemon production. And we have many herbivores that can be pests, but thanks to many natural enemies that we have in the system, we have only a small number of citrus pests. Okay, all this good control is done by many natural enemies, predators, parasitoids. Part of them were introduced at the beginning of the last century, and nowadays they are naturalized, and we have a good system. So, breaking this equilibrium for us is a worry problem, and we are trying to maintain this equilibrium. This is not easy, and as you will see. This is my presentation. I will try to do slow and try to transmit. First, I will explain you the classical biological control that we conducted in Spain for the African citrus psyllid, and then I will explain the strategies and the program and projects that we have conducted with HLB. First, classical biological control of Triotha erythrae. Okay, as presented before, the HLB is transmitted mainly by two psyllid vectors, the African citrus psyllid and the Asiatic citrus psyllid. The African citrus psyllid is Triotha erythrae, which was introduced in Canary Islands in 2002 and in other areas from Portugal, Madeira, at the end of the 90s. Okay? And in Canary Islands in 2002, and the problem appears in 2014 when it was detected in North Spain, in Galicia. Okay? This is not the citrus area for us, but the landscape is plenty of lemon and citrus in the backyards and in the garden. Okay? So Triotha erythrae appeared in this area and invaded the region very fast. Okay? And indeed, colonized part of the Cantabric area, the north of Spain, and also the coast of Portugal region, also very close to the main citrus production. Well, probably you are not familiar with Triotha erythrae. This is the African citrus psyllid. Okay, the adult it's quite different from Diaphorina citri, this aspect. Also the nymphs are different, but the most the different more evident is the damage on the leaves. Okay, produces this kind of bulbs and galls in the leaves, very easy to detect under field conditions. You can see that this was the aspect of the citrus tree in Canary Islands before the introduction of the parasitoid. You will see, and obviously there was not any chemical treatment there. Okay, if you didn't treat with chemicals, the aspect of the citrus were there. Okay, the nymphs are below these galls in the under part of the leaf. Okay, this is the nymphs. So from that we started, apart of other strategies, we started a classical biological control. We knew that in South Africa, where is the origin of this pest, there were some parasitoids. We were there, we established a classical biological control. As some of my colleagues at IVIA visited different areas in South Africa, prospected for parasitoids there. We conducted some preliminary trials there of efficacy, and with the collaboration, of course, of some universities there, we detected 3 species of parasitoids. Okay, Tamarixia tree, this species, and a new species of Tamarixia that was not classified. Well, Tamarixia tree was the most abundant. Tamarixia tree was specific. Tamarixia tree was the parasitoid that was already used in other islands to control Diaphorina, Triotha erythrae. So we conducted all the legal permissions from that our ministry established, specificity trials to confirm, like Nikos explained, that the parasitoid was free of HLB. And after obtaining all the permission and the specificity trials, we started the first releases in the Canary Islands in 2018. Okay, we conducted just one single release point. Okay, at the beginning. Later, we conducted other releases, but with this was just one single release. The parasitoid colonized all the island very fast. Okay. If you want to see the numbers, this is the percentage of orchards infested with Triotha erythrae, and remind the damages that this psyllid produces on the tree. Is very clear to see the damage. Okay, and at the beginning, this is the 3 big islands: Tenerife, Gran Canaria, and La Palma. And you can see that in autumn 2018, the presence of Triotha erythrae, the African citrus psyllid, in those orchards was abundant. Okay. It was common, and it was very easy to detect the psyllid on the tree. Just some months before the first release in Tenerife, the island that I started, the levels decreased dramatically. This was in just 2 years after the release. The success was incredible. Okay, indeed, you can see the percentages of orchards with Trioza erythrae. Trioza erythrae almost disappeared. Okay. For example, we had some projects studying the dynamics, the performance on different cultivars. We had to stop because we couldn't find Trioza erythrae. Okay, and what we found was these holes in the nymphs. The percentage of parasitism was incredible. Okay, I've never seen this level of parasitism. Okay, and now, well, this is from 2020. This is what you can see in the field. You went to the field and you see the old leaves. You see all of them with damages, and the new ones were free, new leaves. Okay. So with these good results, we started the project in the peninsula, in Spain, okay, releasing in Galicia in 2020, okay, and for this, remind that what I told you, the level of the presence of the African citrus psyllid in Portugal and in the north part of Spain, okay. We started with 3 release points. This area is that one, Galicia, in northwest Spain, okay? And from these 3 release points, just after 6 months, the distribution and the dispersal was like this, okay? Again, the results were again very good, okay? And 18 months later, we conducted more releases, the red points, okay, the red you see, and the distribution was very good. Again, the same graph that I showed you for Canary Islands, you can see the percentage of trees with the African citrus psyllid, okay, this is Pontevedra, one of the provinces, again, it was so common, and after 2 years, again, the African citrus psyllid disappeared. Okay? In the other provinces in North Spain, the same. Okay? So, one more time, the success of this classical biological control was almost perfect. And the same picture, this is in Galicia. You can see the new leaves, completely clean of African citrus psyllid, while the old leaves were attacked and with the symptomatology of African citrus psyllid. Okay? And this is very interesting, okay? And hopefully, I would like to occur here the same. This was the picture of the Portugal, the presence of African citrus psyllid in different provinces in Portugal in 2023. This is in 2025, okay? And the legal order that the Ministry of Agriculture, okay, I'm going to translate this paragraph, okay? They have considered eradicated the African citrus psyllid in the south part of Portugal thanks to the action of Tamarixia tree, okay? This is very interesting, and for us, from Spain, this was perfect because this part is where the citrus industry starts in Spain, okay? So we have avoided this risk at the moment from the African citrus psyllid. Okay, so this is an example with African citrus psyllid. Now we are more or less comfortable with this situation. It's very difficult to find Trioza erythrae in the Iberian Peninsula. It's very difficult to find the African citrus psyllid in Canary Islands, okay? But, unfortunately, as Nikos and Melenaos presented, Diaphorina citri was detected in Israel and in Cyprus in 2023. And for us at the moment, this is our big threat, okay? So we started a collaboration with the Cyprus government, with the research centers of Melenaos also, and we try to assist in the classical biological control program that you started with Tamarixia tree. We have conducted here 3 different missions. I will try now to summarize the main results that we obtained from these collaborations. Okay, Probably you remember Alejandro Trena, who conducted the first mission together with Meritxell, okay? Myself, I was here in March 2024, and Alejandro again in July 2024, okay? So this was the first situation that we found in November 23, okay? We visited several infested areas, okay? Together with some of you, okay? And we collected some samples of the Diaphorina in order to know the origin of the populations, okay? Now we have some results with this. We conducted some molecular analysis from the COI and phylogenetic analysis, and I'm not going to present all these letters, the nucleotides, etc., but what is very interesting is that all the samples that we collected here, all the 24 sequences that we analyzed, all of them were 100% identical, okay? And identical to the Israeli populations that we included in the study, okay? So probably the population is the same, the Cyprus and the Israel, okay? So until now, this homology gives the conclusion that probably there is only one single introduction, okay? And from this introduction, it occurred a rapid island invasion, right? More interestingly, we also conducted this phylogenetic tree. Now it's improved, it's not the last version, but to understand very easily this, there are two clades, there are two big populations in the world, okay? It seems that the population from Cyprus has an Asian origin, okay? Indeed, with other sequences that we have tested, the Cypriot population is identical from one population from southwest China, okay? So different from the California and Florida. Well, in March 24, we came back again here with the main objective to know how Diaphorina citrus spend the winter, okay? We were very interested in this because our winter in the Mediterranean area is different from Florida, Brazil, other areas where we had experience, and we wanted to know that. For this we visited, again with some of you, some of the areas of field orchards and also some urban gardens, and what we observed, that is the same that Melenaus presented before, is that Diaphorina citrus survive the winter as adult, okay? And this is very important because this can have some implication for the management. There is a cohort very well established coming from winter and laying the eggs from the spring flushes, okay? So at that time in March we only found adults and eggs in the small flushes, okay? So there is a synchronized cohort that I think now is the same situation that you are finding at the moment in 2024, okay? Well, we conducted some measures, but this is just a graph that we conducted, okay? These were two orchards from Limassol, very close from each other, just 1.5 kilometers, I think. And in one orchard, the grower didn't apply anything, okay? So it was around a percentage of 67% of presence on the flushes. And in this orchard, the grower applied lambda-thialotrin and pyrethroid just 2 weeks before the sampling. And you can see that the flushes were free. And I'm sure that this is due to the mortality to the adults that were overwintering in this orchard, okay? So they could kill the adults, okay? Then we also observed, well, this is already you know, that in the south part of the island the invasion was before the northern part, okay? And what we already observed, and this is very important to establish the biological control program, is that gardens and backyards were already infested in March with Diaphorina citrus. And as in Spain, all the area is infested of citrus. So this is good for us, but it's also very good for Diaphorina citrus, okay? Well, and now about the parasitoid. This is the results that we had at the moment. These are coming from the third mission. At the moment, the result that we had, and this is the report that we sent to the ministry, is that the parasitoid was released in March, May in 4 different locations, probably 8 that Nikos said, but we had that were 4 points of release. And we wanted to know and to check if the Baryxia was established and to collect the parasitoids in order to molecularly analyze to confirm that the parasitoid was the same that was released. Okay, so Alejandro visited 17 citrus orchards and checked for everything, and this was the situation that he found. Okay, Diaphorina was present along the island in 15 of the 16 orchards that he visited, and in 12 of those 15 orchards, Tamarixia was present. Okay, we are talking about July 2014, 24, sorry. Okay, and the parasitoid was released in April, May, sorry, 2014. So you can see that some areas were already with parasitized nymphs. So for this, indeed, as I said, in 2 orchards, we found parasitism in spring flushing. That means that we visited the orchard, and you can see easily the flushing is coming from the spring, because already old leaves, and the new flushing. So we separating the 2 flushes, spring and summer, and we detecting parasitism already in spring flushes. That means that this parasitoid is already present here before the release. Okay, so this suggested a possible native parasitoid already present in the island. So we collected some of the parasitoid. Later, Nikos sent us other new samples to molecularly analyze. At the same with the COI methodology, we conducted this analysis, and we confirmed that this parasitism found in spring was totally different, this parasitoid, from the Tamarixia radiata released in April, May. Okay, of course, the Tamarixia radiata was also present and was the same strain that in California was being released. Okay, and all the wild sequences were 100% identical, and this support that there is a consistent native population. So this is very interesting now, because it's the first evidence that a native established species occur in Cyprus, a Tamarixia species. As far as we know, it's a black color, okay, different from the female of Tamarixia radiata that has a more yellow color, and you can see in the phylogenetic tree that is far away from Tamarixia radiata. Okay, indeed, farthest than Tamarixia tree. Okay, so we characterize molecularly the new species. We also sent to some colleagues taxonomists, and nowadays I can say that already there is a name for this new species for citrus. Okay, and our colleagues Emilio Guerreri and Kristen Hanson classified as new species, and the name probably is Tamarixia citricola. Okay, so this is very interesting. Okay, because there is a new picture, a new view to continue with the biological studies. Now it's very important to know the establishment of this species and how it's going to collaborate with the introduced species, which is already established in the field too. Okay. Well, now some words to explain you other works that we are doing at IVIA, my institute, okay, against Diaphorina citri and against HLB. We have a huge program trying to identify tolerant citrus plant genotypes. Later I will explain you some ideas, but what is important is that we have a rootstock breeding program more than 40 years established at IVIA, okay, and we already evaluated more than 500 hybrids, okay, from some of the tolerant or semi-tolerant rootstocks, Poncitrus, Nicotitrus, and Ethmocitrus, and we already have some rootstocks that could be interesting. This is the oviposition of Triotha in different rootstocks. Okay, Citrange carrizo is the rootstock more used in Spain. Okay, and you can see it's a very good host for the African citrus Psyllid. And all the rest of plants, except this, that is a South African one, okay, are rootstock that we have selected based on defenses and things like that, okay. But you can see that we have some promising rootstocks. Obviously, they are not going resistant at all, okay, but at least could work better than the one that we have. And this is also, we also analyze some of these rootstocks compared with the American ones that they are using, for example, in Florida, the US 942 and others. Okay, this is the carrizo that we use as a control. As you can see, that these two rootstocks are more robust against Diaphorina citri. We have some experiments in Brazil and in Florida with these rootstocks, okay, because now in Spain is being planted for Alcaide 5 a lot, so this is very interesting, and this rootstock is also very good for other pests like mealybugs and for spider mites. So this is also to take into consideration for those of you who are going to plant new plantation, and you have to select the rootstock, okay? This is not going to give you the solution, okay, but at least can give you a help managing this. Indeed, you can see, well, we have many results on that. This is just the selection of the comparison between Citrange carrizo, that is the rootstock that is most used, and the new one that we are promoting, Cativia, for Alcaide 5. As you can see, that is more attractive to both parasitoids, Tamarixia erbui and Tamarixia radiata, than the Citrange carrizo. And how can you explain this? But very easy. We have conducted some transcriptomic and volatoloma analysis, and we have seen that this rootstock has a higher biosynthesis of secondary metabolites and more volatiles that attract the parasitoids. Okay, so there is some science behind this, and where we are working with the rootstocks very hard. And then another research line that we are working is the enhancement of citrus defense mechanisms. Okay, I'm just going to give you 2 words on that. The idea is to, thanks to some defense induces, we want to fortify the citrus. Okay, we have some ideas. Okay, we have some small molecules that can activate some plant hormonal pathways which are related with the defenses of the plant. We have some volatiles that communicate with the plant that are able to activate the defenses, okay? And indeed, using some of these small molecules and also the volatiles, we have obtained similar results that, for example, gibberellic acid that is promoting in Florida and other antioxidants applied against HLB can obtain. Okay, so we are dealing with this. We have also selected some genes that are able to activate these antioxidant pathways, and now we are working in 2 lines. One, to select those cultivars and those rootstocks that have more activated these genes that are able to activate the expression of the oxidant. And another research line is the role of the jasmonate pathway acid. We were working with this gene, the MIG2, okay, and we were very surprised 2 weeks ago with a new paper from China. Maybe you are aware that they talked for the first time about the resistance of HLB modulating the MIG2. Okay, so we are working in the same line. Okay, trying to stabilize this gene, which is a transcription factor. It's a gene that can modulate these defenses, the mediated acid. And as I told you, we are working for trying to select cultivars and roostocks with a more with this higher amount of meat, okay, and also with the CRISPR trying to edit different genes that are able to resist the HIV, okay. This is for the future, but the small molecules, the elicitors, and other plant defense inducers, this is the present, okay? We have some solutions, and now we are testing some of them, okay? This is just some ideas in what we are involved. And with all of this, if you have any question, I will be more than happy to solve your doubts. Thank you.
Είναι πολύ ενδιαφέρον αυτά που έχουμε ακούσει για τους βιολογικούς παράγοντες. Από ό,τι αντιληφθήκαμε, δεν είναι μόνο ο βιολογικός παράγοντας που έχουμε εισάξει εμείς στην Κύπρο, αλλά και ένας πρόσθετος βιολογικός παράγοντας που ήδη υπήρχε και μας βοηθούν οι συνεργάτες μας να τον ταυτοποιήσουμε και να δούμε πώς μπορούμε να αξιοποιήσουμε όλο αυτό το καθεστώς υπέρ μας. Αλλά και ακόμα αυτά τα ανθεκτικά υποκείμενα που βλέπουμε να είναι καθοδόν, να μπορούμε να δούμε πώς μπορούμε ακόμα καλύτερα να τα αξιοποιήσουμε για να χτίσουμε ένα ισχυρότερο τείχος ενάντια του βακτηρίου που προκαλεί την ασθένεια του κίτρινου δράκου. Σε λίγο θα βγούμε για ένα μικρό διάλειμμα, αλλά πρωτού βγούμε, μιας και έχουμε εδώ τον Alberto με τα θέματα αυτά που είναι φρέσκα στο μυαλό μας, παρακαλώ, είμαστε στη διάθεσή σας για οποιοδήποτε ερώτημα. Μπορείτε να το κάνετε είτε στην ελληνική είτε στην αγγλική γλώσσα, υπάρχουν οι διερμηνείς, για να μπορούμε να πιάσουμε την απάντηση που χρειαζόμαστε. Αν έχουμε κάποιο μικρόφωνο, εταιρεία πίσω που μπορούμε να κυκλοφορήσουμε για να πάρουμε τις διάφορες απορίες, ή αν έχετε και απορίες για τους προηγούμενους ομιλητές μας, τον Νίκο, τον Σεραφίδη και τον Μέλενα.
Μπορούμε να αφήσουμε όλα στο τέλος τις ερωτήσεις.
Όπως νιώθετε. Προτιμάτε να τα αφήσουμε όλα προς το τέλος? Βεβαίως, ωραία. Παρακαλώ, έχετε κάποιο ερώτημα? Ένα λεπτό. Μπορούμε να έχουμε ένα μικρόφωνο? Εδώ. Σας ακούμε.
Από ό,τι αντιλαμβάνομαι, μεγάλες οι εξελίξεις αυτά τα πράγματα.
Ένα λεπτό, σας παρακαλώ, για να βάλει το... You don't know how to use that?
Από ό,τι ακούσαμε σήμερα, μεγάλες οι εξελίξεις στη γεωργία για καταπολέμηση των διαφορών ασθενειών. Από εμάς τους παραγωγούς τι ζητάτε να κάνουμε? Διότι εμείς είμαστε οι παραγωγοί, εμείς ασχολούμαστε στα χωράφια, στα περβόλια. Ποια είναι η ηθεσία που πρέπει να πάρουμε από εσάς? Τι πρέπει να προσέξουμε, τι πρέπει να κάνουμε? Και δεύτερο, κίτρινος δράκος, πώς θα είναι πήρε του τόνο?
Okay, yeah. Okay, I will start with the second one, okay? I think that one gloom being means yellow dragon, and this is because the citrus tree suddenly appeared one yellow branch that is due to the bacteria which blocks the sapia of the tree, and the branch is yellow, and it looks like, according to the Chinese, like a dragon. I think this is the reason. Okay, and the first question was related to what the growers can do. Okay, this is my personal view. Okay, at the moment, what you have to do is to maintain the populations of Diaphorina citri low. Okay, but for this you have to follow the recommendation from the administration. Okay, to treat aggressively with non-selective pesticides, this is a risk at the moment, because according what occurred in other citrus area, the main solution that you can rely now is biological control. Okay, with the Tamarixia radiata that is introduced from California. Okay, so for this you have to respect this natural enemy, and you have to protect and to conserve the parasitoid. Okay, This is very important. I'm sure that later my colleague Mark will explain what in California they are doing to do that, okay? But you have to take into consideration that diaphorina citrate by itself is not a huge problem for the citrus, okay? So you can manage, or you can live with small populations, because at the end this is like an aphid, okay? You can maintain slow populations while biological control is conducting the work, okay? I suggested, for example, to do winter treatments before the flushing, okay? Because as I saw last year, treating the adults that are overwintering, okay, they can reduce the first flushing, okay? So in commercial orchards, at the moment, I would treat selectively, okay, and in the gardens, backyards, etc., I would increase the populations of the natural enemies that at the end will be your solution. What it's sure everywhere is that chemical control is not the solution, okay? This is for example, in Brazil, they relied the management of diaphorina and HLB on chemical treatment. They haven't succeeded. Okay? Now they have a lot of problems with HLB because appears the Asiatic strain of HLB, and now they have resistance with the pesticides. They treat the pesticides as not effective as before, so chemical is not the solution. So at the moment, you still have to rely with biological control and to maintain the low population with selective pesticides. Selective pesticides, at the end of the day, for our American colleagues, we have 1, 2, 3 products at the end for the European restrictions, so we have to play with this. Okay?
Νομίζω με κάτι. Πώς.
It's okay, it's okay. No, it's.
Ούτε. Νομίζω με κάλυψες, Αλμπέρτο, βασικά στην ερώτηση. Και εγώ αυτό θα έλεγα. Οι οδηγίες του τμήματος γεωργίας προς τους παραγωγούς είναι όταν βλέπετε να αυξάνονται οι πληθυσμοί της ασιατικής ψήλας, να προχωρήσετε κανονικά τους ψεκασμούς βάσει των οδηγιών που γράφουν τα συγκεκριμένα φάρμακα, τίποτε περισσότερο. Το τμήμα γεωργίας, όπως σας είπα, σε συνεργασία με το Ινστιτούτο Γεωργικών Ερευνών, θα ξεκινήσει σιγά σιγά και πάλι την εξαπόλυση του βιολογικού παράγοντα. Να μην σας ανησυχήσει αν δεν ερχόμαστε συγκεκριμένα στο δικό σας αγροτεμάχιο για να κάνουμε αυτή την εξαπόλυση. Επιλέγουμε συγκεκριμένα τεμάχια έτσι ώστε να γνωρίζουμε αν μπορεί το έντομα αυτό το παράσιτο να πολλαπλασιαστεί, και αφού πολλαπλασιαστεί και δράσει ικανοποιητικά, τότε αυτό από μόνο του θα προχωρήσει σε διάφορες καλλιέργειες και παντού. Οι εξαπολύσεις μας είναι συγκεκριμένες, είναι μικρές, είναι στοχευμένες. Ωραία. Και αυτό ναι, κινείται και θα προχωρήσει παγκύπρια. Άρα αυτό να μην σας ανησυχήσει. Και προσπαθούμε εμείς με τα δικά μας τα μέτρα να αυξήσουμε το βιολογικό παράγοντα σε τέτοιο βαθμό, να διατηρείται σε τέτοιο πληθυσμό που να κάνει από μόνο του τη δουλειά του και εσείς σιγά σιγά να μην παρατηρείτε τον διαφορίνα σύντρι. Κατάλαβα, να μην το βλέπετε. Αλλά όντως, εάν εμφανιστεί σε ψηλό πληθυσμό, τότε ναι, θα χρειαστείτε εσείς τη χημική καταπολέμηση. Άρα, σε κάποια φάση, αυτό που προσπαθούμε να πετύχουμε είναι να αποφύγετε εσείς να ψεκάζετε περισσότερο, εντάξει, γιατί θέλουμε το βιολογικό παράγοντα να ξεκινήσει να κάνει σωστά τη δουλειά. Σας λέω ότι να σταματήσει να ψεκάζετε, αλλά μόνο υπάρχουν και άλλοι εχθροί και άλλες ασθένειες στα σπίτια, το γνωρίζουμε. Απλώς θέλω να ψεκάζουμε και να χρησιμοποιούμε τα χημικά σκευάσματα σωστά και περιορισμένα όπως πρέπει.
Πρέπει να μας υποδείξετε τι φάρμακα είναι για τούτο σκοπό.
Τα φάρμακα μας υπάρχουν στη ιστοσελίδα μας γραμμένα και να βεβαιώσουμε το επαρχιακό γεωγραφείο ότι θα τα έχετε συγκεκριμένα εσείς να μπορέσετε να γνωρίζετε ποια είναι αυτά.
Συγγνώμη.
Ένα λεπτό σας παρακαλώ, γιατί έχουμε και ξένους. Ναι, ανοίγουμε. Δεν ξέρω πώς το δουλεύουμε. Μιλάτε να δω.
Συγγνώμη, αυτή την εποχή τώρα τα δέντρα μας είναι φορτωμένα με φρούτα για χυμοποίηση. Παγέτσια, ομφαλοφόρα και χρειάζονται να τα ψεκάσουμε για τη μεσογειακή μήλα. Τι είναι, σαν εγώ χρησιμοποιώ το κάσιο για τη μεσογειακή μήλα. Πειράζει αυτά τα ωφέλιμα έντομα που θα τις απολύσουμε να καταπολεμήσουν αυτούς τους εχθρούς που μας λέγεται τώρα.
I don't know how you treat against the medfly, but in Spain, my experience is to treat the medfly with bait treatments. This is very selective to natural enemies because you only treat part of the tree, the sunny part of the tree with the bait. Okay? To do complete treatments against the medfly is not recommendable for this problem because you are killing everything. And probably if you treat with pyrethroids, I don't know, I don't follow the name of the product, but anyway, you have pyrethroids or spinosad, okay? Lambda cyhalotrina, spinosad, and tau, these two solutions, okay? To treat with the pyrethroids the full orchard without using bait treatment, this is crazy. This is crazy because you enter in the fireball, I don't know, in the routine to treat because you are going to kill predatory mites, you are going to kill parasitoids, and then you will have to treat against mealybugs, wildflies, spider mites. So my advice would be to treat with bait treatments, or both with the spinosad or with the pyrethroid, okay? And only the sunny parts of the tree, okay? For the medfly, for the Ceratitis capitata, okay? Did you follow me? And I forgot to say one thing, it's about the roostocks. When I visited the Cyprus industry, okay, I saw different proportion of roostocks, okay? This is one thing that if you are going to do new plantations, you have to take into consideration. The selection of the rooststock is key for pest management, okay? Apart of other characteristics for the soil, the water, etc., okay? And this is one of the reasons we are excluding all these rooststocks, okay? Citrange carrizo, for example, that is the most used in Spain and other citranges, nowadays the plantation is very limited, okay? We are trying with Fornera alcaide 5, that is very resistant to pest and also draw and other things and other plants. This is very important to study your soil, to study your conditions, and of course only planting certified plants, that is obviously followed by the Cypriots. Okay?
Κάποιο άλλο ερώτημα?
Ναι. Ευχαριστώ.
Θέλεις?
Ευχαριστούμε. Είναι πολύ εντυπωσιακή η δουλειά που έχετε παρουσιάσει, η δουλειά που κάνετε, η οργάνωση και η εντυπωσιακή δουλειά που έχει ξεκινήσει να γίνεται στην Κύπρο. Εγώ ήθελα να ρωτήσω σε σχέση με τον πληθυσμό του παρασιτοειδούς που έχει εισαχθεί, κατά πόσο θεωρείτε ότι η εισαγωγή ενός μικρού πληθυσμού, μικρού αριθμού ατόμων, ενδεχομένως να δημιουργήσει προβλήματα στην επιβίωση του, από πλευράς γενετικής παραλλακτικότητας. Κατά πόσον είναι απαραίτητο να επανεισάγονται νέοι πληθυσμοί για να ενισχύεται η γενετική ποικιλομορφία του είδους, του πληθυσμού στην Κύπρο. Ευχαριστώ.
Okay, I see your question, and I'm going to wait until the next talk, okay? Because, no, I mean, the populations that you have introduced here in Cyprus is already after a genetic studies and different tissue lines that at the end is very powerful regarding this genetic background, okay? So, I think that the numbers that Nikos and David and Mark are working, they are already good numbers to avoid this. But take into consideration that this is coming from a previous selection. So the genetic, I think, will be a problem, but I'm sure that Mark will explain you better than me. Okay? But sometimes this is, for example, from South Africa, we introduced 7 isolines. Okay? We crossed the isolines, okay? And we released, the first release that I showed you was just 40 adults. And from these 40 adults, it was delayed. Okay, so this confirms that the selection that you have done here in Cyprus is correct, because you are releasing more than 40, for example, and the number of isolines I think is 17, okay, and we only conducted 7 from South Africa, so it's more powerful now, the system here, okay.